Welcome students in the online organic chemistry lecture series. Today we are going to study an important topic in the biomolecule chemistry, and that is nucleic acid, and which is also the part of your organic chemistry syllabus in semester 6 TYBSc class. Now this nucleic acid chapter is a very short chapter actually. Only a few concepts are there, but they are very important while learning the organic chemistry. And the name of that topic is nucleic acids. Now we know that these are the macromolecules present in the living organism. I can say nucleic acids are the macromolecules. Macro means big molecules. I can say present in the living organism. Maybe you take a plant cell or animal cell. These nucleic acids are there. Now every living cell contains conjugated proteins, and that are known as nucleoproteins. Okay, and you know very well DNA and RNA are the important nucleic acids which are present in the living cell. Okay, so DNA and RNA is the fundamental life material of the living cell, we can say. Okay, now what are features of these Lewis acids? They are colorless, they are water soluble solids. Okay, they have high weight because these are the macromolecules. Also we can call them as a biopolymers. Mainly they contains carbon, hydrogen, oxygen, nitrogen and phosphorus as the main elements. Okay, and they can be soluble in dilute alkalis, but they are insoluble in dilute acid. Now the basic backbone of these protein molecules is a polypeptide chain, and the polypeptide chain is made up of three components. One is base, another component is sugar, and third component is a phosphate. So when base, sugar, phosphate, base, sugar, phosphate, in this way a nucleic acid strand is formed there, or we can say polypeptide chain is formed there. So this is the basic uh, chain which is there in the nucleic acid, we can say. Okay, so this is the introductory part of this nucleic acid. Now we'll see nucleic acids are classified depending upon the nature of sugar present in it. Okay, you know, basically two nucleic acids are there, DNA and RNA, and both these two are classified depending upon the nature of sugar which is present there. Okay, so we know that this RNA is a single stranded material and DNA is a double stranded material. So DNA will stand for deoxyribonucleic acid. Why it is called as? Because it contains a deoxyribose sugar. Okay, and it is found in the nucleus of the cell. So the two major differences are there in DNA and RNA. One is that it contains deoxyribose sugar, DNA contains deoxyribose sugar, and it is found in the nucleus of the cell, where RNA stands for ribonucleic acid and it contains ribose sugar. Okay, and it is found in the cytoplasm of the cell and not in the nucleus of the cell. Now these are the two sugars which are present in DNA and RNA. So as I said earlier, DNA contains this ribose sugar. So this is the structure of this deoxyribose sugar. So you can see here it is a 5-membered oxide ring. Okay, this first carbon is ionomeric carbon, as you know, and it has OH group above the plane of this ring. Okay, and there at 2 position there is a absence of OH group. You see here at second carbon there is no OH group, so it is known as 2-deoxyribose. While in ribose sugar at 2 carbon there is a OH group. Rest of the structure is the same, and if you give the numbering, then you will find here that this is carbon number 1, 2, 3, 4 and 5. So this is 5 dash carbon end and this is 3 dash carbon end, remember. Okay, so these are the differences between the sugars which are present in DNA and RNA. Now we'll move further. Now if we do the hydrolysis of nucleic acid, then we get nucleosides, nucleotides, and further hydrolysis will give only three components: sugar, base and phosphate. So you see here basically nucleic acid on hydrolysis in alkaline medium like aqueous ammonia or barium hydroxide at 115 degrees Celsius, or in living cell generally this hydrolysis of nucleic acid will be carried out by the enzymes. Okay, and the name of that enzyme is nuclease which does the hydrolysis of nucleic acid, or which break down the nucleic acid, I can say. So nucleic acid on hydrolysis first of all give nucleotides. Okay, nucleotides on dephosphorylation gives nucleoside. That means if we compare the nucleotide and nucleoside, then there is a difference of only this phosphoric acid. H3PO4 is present in nucleotides, whereas in nucleoside there is a no H3PO4. And if we further hydrolysis this nucleoside in presence of inorganic acid, you will get sugar and bases. So what we can say, the basic component of nucleic acids are sugar, bases, and this phosphoric acid, I can say. So these are the three basic components of the nucleic acid. So from which the DNA and RNA is made up of. Sugar, base, and phosphoric acid are the main components of the nucleic acid. Okay, you can say. Now we'll move further. Now let's see one by one. Now first of all we'll see sugar component and basis component. Now these sugar components, just we have discussed about that these are the 5-member oxide rings. This is carbon number 1, this is carbon number 2, 3, 4 and 5. So at carbon number 2, if there is absence of OH group, then it is known as 2-deoxyribose sugar. Okay, and if at 2 carbon there is a presence of OH, then it is known as ribose sugar. So these are the two kinds of sugars that are present in DNA and RNA, remember. So particularly in DNA the 2-deoxyribose sugar is there, and in RNA there is a ribose sugar. So this is the main difference between DNA and RNA material. Now what kind of bases are there, that is also very important. Generally the bases are of two types. One is a pyrimidine derivatives. Okay, so you see here pyrimidine, this ring is a pyrimidine ring. So there are two pyrimidine bases, one is cytosine and another one is a thiamine, remember. So you can remember C and T as a pyrimidine bases. Okay, and again if we have a purine ring into the bases, then they are known as purine derivatives. So two purine derivatives are there, adenine and guanine. So these are the four bases particularly present in the nucleic acid. So cytosine stands for C, thiamine stands for T, adenine stands for A, and guanine stands for G. So these are the four main pyrimidine and purine bases which are present in the nucleic acid. So these sugars and bases are the basic components of the nucleic acid. Now we'll further see. Now if you compare the nucleic acids like DNA and RNA, now combinedly both DNA and RNA made up of uh, adenine, guanine, cytosine. So these three bases are common in both DNA and RNA. Remember, in DNA and RNA, these three bases are common: adenine, guanine, and cytosine, and phosphate group is also common. But if we compare the differences, then in DNA, thiamine base is present, and in RNA, instead of thiamine, there is a uracil base present. So this is major difference, one of the major difference present between DNA and RNA. So once again, I will explain. In DNA, thiamine base is present, and along with thiamine, these three bases, adenine, guanine, and cytosine is there. Okay, but in RNA, along with these three bases, uracil is there. There is no thiamine. Remember. Okay. Then another difference is the sugar which is present in DNA is 2-deoxyribose sugar, right? And whereas the sugar present in RNA is ribose sugar. So in this way we can distinguish between the components of DNA and RNA. Okay. Now I think it is very simple to understand. Now let's move further. Now nucleosides. Just before few slides, we have seen about the hydrolysis of nucleic acids, and there we have seen that nucleic acid is generally hydrolyzed into nucleotides and nucleosides. Okay, and nucleoside further hydrolyzed into sugar and bases. So what is that nucleoside, that we have to see first now. Okay, so nucleosides are the sugar based compound. Remember, it is very simple. Just we can remember it like sugar and base compounds in the nucleic acid is known as nucleoside, or sugar plus base part of the nucleic acid is known as nucleoside. Remember, so just they are formed by the combination of sugar, or it may be ribose or deoxyribose sugar, okay, with base like purine and pyrimidine derivatives. And again we can say nucleosides are the glycosides, and which are non-reducing. So nucleosides containing pyrimidine bases are much more stable to hydrolysis as compared to nucleosides with purine bases. So this is important thing you have to remember. Now let's see the example of nucleoside. Now you see here, this is a sugar component, okay, and this is a base component, okay. So what is the name of this? This is adenosine, because adenine is with the base. Similarly, this is another example. This is known as deoxyadenosine. Okay, so the difference between adenosine and deoxyadenosine is just the sugar component is different. Here there is a ribose sugar, and here there is a deoxyribose sugar. Then third example is a guanosine. Remember, and the next example is deoxyguanosine. Here also the difference is only in between the sugar component, the base is same, okay. So important thing to remember is that in nucleoside, the C1 of the sugar is bound to one of the nitrogen atom of the base, and the linkage is known as a glycosidic linkage or beta linkage. So this is very important parameter which you have to remember, that in nucleoside there is a glycosidic linkage. Now in which atoms this linkage is present, so you remember that this carbon is C1 carbon, okay. This is a C1 carbon, right? This is a C2 carbon. So the base is attached to sugar with this C1 carbon. So C1 carbon is attached to the nitrogen of the base. Here also C1 carbon is attached to nitrogen of the base. In guanosine also C1 carbon is attached to nitrogen of the base. In deoxyguanosine also C1 attached to nitrogen. So we can say that in nucleosides, the C1 of the sugar is bound to the one of the nitrogen atom of the base, and that linkage is known as a glycosidic linkage, or it is also known as a beta linkage. So this is about nucleosides. These are just sugar plus base compounds. So sugar and base are attached with each other by means of glycosidic linkage, I can say, and they are non-reducing. Okay, so this is about nucleoside. Now let's move further. Now let's see about nucleotides. Okay, so as I said earlier, the difference between nucleoside and nucleotide is only in the components. Nucleosides have only sugar plus base component, while in nucleotides along with sugar and base there is a phosphate group. So this is very important. So remember, nucleotides are made up of three components: a phosphate group, sugar and base, okay. So these are the three parts of nucleotide. See, this is a base, the name of this base is adenine, okay. This is a sugar, this is deoxyribose sugar, and this is a phosphate group. So this can be called as a nucleotide, okay. So again the same structure is there. So if such a kind of nucleotides joins with each other, say two nucleotides, three nucleotides, four nucleotides, and so on, thousands of nucleotides, if joins together, then there is a formation of polynucleotide strand. Remember, so one nucleotide joins with another nucleotide, okay, then that nucleotide joins with another nucleotide. In this way there is a chain of nucleotide, and that chain is known as a polynucleotide strand. Remember, so this is the basic difference between nucleoside and nucleotide. Nucleoside have only base and sugar, while nucleotide has base, sugar as well as phosphate group. Now this is a very important, that a nucleotide is a backbone of DNA and RNA. We can say it is a building block of DNA and RNA, okay, and they have functions like cell signaling, metabolism, enzyme reactions, okay, and three parts, phosphate group, 5 carbon sugar and nitrogenous bases, okay. The four nitrogenous bases in DNA are adenine, cytosine, guanine and thymine. Remember, so these are the four bases which are present in DNA: adenine, cytosine, guanine and thymine. While in RNA, instead of thymine there is a uracil. That is the only difference. Okay. Now a nucleotide within a chain makes a genetic material of all known living things. That means we can say that in any living cell there is a nucleotide, and from that nucleotide there is a formation of genetic material, and that genetic material will be either a DNA or it will be a RNA. So they also serve a number of function outside the genetic information storage. Also they are the messengers of messengers also, they act as a messenger as well, like mRNA, and they are also energy moving molecules. So they have a lot of difference. So we can say that without nucleic acid, life is not possible. So every living cell, every cell has a nucleic acid in the form of DNA and RNA, and that DNA and RNA is made up of nucleotide. Nucleotide is made up of nucleoside, and nucleoside is made up of sugar and base. Okay, so the three basic components, sugar, base and phosphate, from which all nucleotides are made up of, remember. Okay, now let's see the primary structure of the nucleic acid. So this is a primary structure of nucleic acid. So how the two nucleotides are joined together, that is shown over here. Now you see here, this is a one nucleotide, we can say. This is a sugar, deoxyribose sugar, which is attached to guanine base. Okay, and the linkage between base and sugar is a glycosidic linkage that is attached to C1 carbon. Then here the 5 carbon is attached to phosphate, and 3 carbon is attached to another phosphate of another nucleotide. So this is one nucleotide which is shown over in this block, and this is another second nucleotide, this is the third nucleotide. In this way, thousands of nucleotide molecules joins together to form a polynucleotide strand, remember. So here also the schematic diagram is represented. So one is a 5 dash end and another one is a 3 dash end. So this is one of the strand of nucleotide, I can say. This is a representation of polynucleotide, I can say. So see here, this circle represents a phosphate group which is at 5 carbon. This represents the 5 member ring, that is a sugar, and these alphabets represent the base. So again another nucleotide, this is another nucleotide, this is another nucleotide. So in this way, many nucleotides joins together to form a polynucleotide strand, and that polynucleotide strand has a two ends, 5 dash end and 3 dash end. Now another important point which you have to remember is that the two nucleotides are joined together by a phosphodiester linkage, remember. So the linkage between the two nucleotides is a phosphodiester, okay, and the linkage between sugar and base is a glycosidic linkage. So in nucleotides, two kinds of linkages are there, remember. A glycosidic linkage which is in between C1 carbon and base, okay, and another important linkage is phosphodiester linkage which is in between 5 dash carbon and 3 dash carbon, remember. So this is the basic core of nucleotide, or this is the basic components of polynucleotide strand, I can say. Okay, so let's move further. Nucleotides are the building blocks of nucleic acids, and you know they are made up of two components, that is a base and sugar, like this. So in nitrogenous bases are adenine, cytosine, guanine, thymine or uracil. So this is repetition, that is I have already explained in previous slide. So though I am once again explaining that the bases present in the nucleic acid are adenine, cytosine, guanine, thymine, and instead of thymine there is a uracil in RNA. That is the only difference. Okay, DNA and RNA each have a 4 possible nitrogenous bases. Only there is a difference between DNA and RNA in thymine and uracil. That is the only thing. Then each of these 4 bases has a different bonding properties, remember. So this is very important. Each base has a different bonding properties. So that's why what is happening, there is a no mix up in one letter to another. Okay, and thymine and uracil probably have almost identical structures and properties, and which is allowing them to fulfill the similar role in two different types of nucleic acid, like thymine in DNA and uracil in RNA. Then a sugar phosphate backbone which allows the nitrogenous bases to strung together, and each nucleotide sugar can link to another nucleotide phosphate to become a single molecule. So that I have explained in previous slide, that is a phosphodiester linkage is present in between the two nucleotides. So that is explaining here. Then when many nucleotides are joined together, then the angle of this phosphate sugar bond most often becomes in such a way that there is a helical like structure in development. Okay, and that is why the DNA, which is a two stranded and naturally takes a shape of a double helix. Okay, the primary structure of nucleic acid just refer to the sequence of its nucleotide bases. Okay, and the way these are covalently bonded to each other. and the sequence of letters of the strand DNA and RNA, and then it is a part of its primary structure, and as is the helical or double helical shape, we can say. So this is some basic information about the structure of nucleic acids like DNA and RNA, I can say. Now we will see, let's in detail about the secondary structure of DNA and RNA. So first of all, we'll see a simple, that is RNA. Now DNA and RNA are chemically similar, only there is a difference in the sugar and one of the base, that is thymine and uracil, that we have seen already, and there is a difference in size as well. Remember, the molecule of RNA is smaller and it is found in the outside of the nucleus, so that is also the important point to remember between the difference between DNA and RNA, that RNA is smaller and it is found outside the nucleus. Then another important point is that the average molecular weight of RNA is ranges from 20,000 to near about 20 lakhs. Okay, so the weight of RNA is also very big, but it is less as compared to the DNA, I can say. Then the X-ray analysis of the RNA molecule exists as a single strand, okay, and which contains the helical segment stabilized by the hydrogen bonding. So if you do the X-ray analysis of RNA, then it is observed that RNA is a single stranded molecule, but it is also in helical form and it is self-complementary, so it doesn't require another strand to stabilize. So here is the pictorial representation by which we can explain the secondary structure of the RNA. So you can see here that this is a single strand over here. This is a single strand of RNA. So this G and C are the nucleobases. Here also C, G, A, U. These are the bases. This part is a helix, and this is a sugar phosphate angle which is developed here. And these are the bases which are present in RNA: cytosine, guanine, adenine, and uracil. So you know there is a uracil base in the RNA instead of thymine as like in DNA. So this is about the secondary structure of RNA and somewhat information about the RNA. Now let's see a DNA, double helix structure, which is also very important and easy to remember. The molecule of DNA is very large, first of all, as compared to RNA, and with a molecular weight up to 150 billions. So you can imagine how big it is, and the length is up to 12 centimeter when stretched out. So you can imagine the DNA is present in the nucleus, and nucleus size is very small, in the range of near about 10 to the minus 13, and in that a small nucleus the DNA is present, and when that DNA is stretched out it becomes 12 centimeter. So what a vital thing it is actually, okay, and they are found in the nucleus, that we know. And in 1953 Wilkinson have done the X-ray studies, and he shown that the DNA have a double helical form, and further we'll see this double helical form. So according to Watson and Crick model of DNA, further after the Wilkinson, the Watson and Crick were the two scientists who proposed the model of DNA consist of two polynucleotide strands coiled right-handed around each other in a double helix. So you can imagine these strand. This is one strand, okay, and this is another strand. So two strands are there. Both are complementary to each other. Both are opposite to each other, okay, and the two strands runs in the opposite direction. That is, we can say if the sequence is C3 to C5 in one strand, then it will be C5 to C3 in another strand. So you can say here is a this is a 3 dash end and this is a 5 dash end, whereas this is a 5 dash end, this is a 3 dash end, like this. So due to this, what happened, the purine and pyrimidine bases in two chain face towards each other, okay, and further the two chains are held together by a hydrogen bonds between the pairs of bases. So you see here there is a this adenine and thymine are in front of each other, then cytosine and guanine are in front of each other. So there is a hydrogen bonding between these two bases. So that hydrogen bonding you will see, for example, if adenine and thymine base is there, then you see the structure of adenine and you see the structure of thymine, then there are possibility of two the hydrogen bonds. So always remember, in between adenine and thymine there are only two hydrogen bonds, remember. While if we see another base pair, that is guanine and cytosine, so if we see the structure of guanine and cytosine, then there are three possibilities of hydrogen bonding as shown over here. So in guanine and cytosine there are three hydrogen bonds. In adenine and thymine there are two hydrogen bonds. So these hydrogen bonds makes this DNA molecule a stable molecule, makes these two stand to attach with each other. So this is a very important parameter of this double helix structure. Other parameters are the distance between these two strands, that is diameter is near about 20 angstrom, remember. Then there are two grooves which are present into this double helix. One is a small groove, another one is a large groove. So small groove is about 12 angstrom unit, large groove is about 22 angstrom unit, and both these two groups constitutes a length of complete one turn, and that is about 34 angstrom unit. And you can see this, that these two strands are opposite to each other. So the word is used here is a complementary to each other. So due to this complementary nature, what happens, the two bases comes in front of each other, and there is a hydrogen bonding between these two bases. So this is all about the nucleic acid chapter. So what we have seen in this chapter, we have seen what is nucleic acid, from which components it is made up of. Okay, so three basic components are there: sugar, base and phosphate. So if you see only sugar and base, then it is known as a nucleoside. If we add phosphate into it, that is sugar, base, phosphate, then it becomes nucleotide. So many nucleotides joins together to form polynucleotide strand. A single polynucleotide strand is there in the RNA. A double polynucleotide strands are in front of each other and complementary to each other in case of DNA. So it is very important chemistry, very important biology which we have seen today, and without which a life is not possible, as you know. So I think this chapter is very much clear to you. So it is very simple to understand. So we'll stop here today. Have a nice day. Thank you.